Hello, biology students. We're on part 3 of Meiosis in Gizmos. We left off way down here on what does the word dominant mean in this particular context with cats. We're going to look at the white spots. The big S genes for spots is dominant over the little s gene, which is no spots. So let's go into Gizmos here and let's look at the spotted colors here. So the white spots in cats are controlled by one gene. It's the S gene. If you get two big S's, you have white spotting, plus whatever color you get on your X chromosome. If you have one big and one little, you're heterozygous for the S gene, you have white spotting plus whatever other color you got on your X chromosome. If you have little s, you're homozygous recessive, then you don't have any white spotting. Now, remember that the spotting gene is on an autosome, not a sex chromosome, so it's not linked to the X chromosome. So now we know about the white spotting. Let's learn about the orange and black coloring. So orange and black is controlled by the O gene that has two alleles. The big O makes you orange, and the little o makes you black. Now, if you're female, you get two O's, and we'll see what happens when that happens. Male cats only get one X chromosome, so they're going to get big O or little o. So males can either be orange or they can be black. They can have white spots as well, but they can only be orange or black. They can't be both orange and black because they only get one chromosome, one X chromosome, and that gene for color is on the X chromosome. Females have two X chromosomes, so they can get two oranges, two blacks, or an orange and black allele, and females can be orange and black because they have two X chromosomes. They can get two different alleles for the color gene. Which of the following fur colors are possible in males? Choose all that apply. Remember, the S gene for white spots is on an autosome, so it's separate from the X chromosome, and the O gene is on the X chromosome, so you only can get one copy of that if you're a male. Remember, males can't be black and orange at any point because they only have one X chromosome. I'll let you fill this one out. The next question asks you what colors are possible in females. So remember, females have two X chromosomes, so they can have one black and one orange. They can have two oranges or they can have two black chromosomes. And they also can have that gene for spotting. So females have a lot more options. Well, at least one more option than males. The appearance of orange and black fur in females is caused by a process called X inactivation. Early in female development, one of the X chromosomes is randomly made inactive in each cell. The X inactivation causes female cells, XX, to have just one active chromosome, just like males, who are XY. Only the active X chromosome will produce fur color. So we can simulate X inactivation here. Here's the active chromosome. Here's the inactive chromosome. X inactivation does not affect the phenotypes of cats that are OO, big O, or little o, because they get two of the same alleles. When one X chromosome is activated in a big O cat, the remaining X chromosome will only just have orange alleles. Same with black. If you have little o, you're black, and if inactivate one of your X chromosomes, it's always going to be black, because black is the only choice you have if you have two little o's. In heterozygous big O little o cats, the X inactivation results in some cells with orange allele and some with black. This gives the cat both orange and black patches. So depending on which X chromosome they inactivate, if the cell inactivates the orange chromosome, that particular cell will have the black allele, the little o allele, and make black fur. If a particular cell inactivates the little o allele, then the other chromosome has an orange, then that cell that's inactivated the black will make the orange color for fur. So here is a good thing to review. Here's meiosis, here's mitosis. You can look for some differences there and review what makes unique cells here. In the meantime, let's go to 28. How many small O alleles does a cat need to have black to show the black fur trait? So if you're black, a black cat, you either have little o if you're a female and no genes for striping, or if you're a male, you have just O because you only have one X chromosome. This is a good chart that summarizes what can happen for colors in cats. Tortoiseshell is just orange and black, and calico is orange and black with white. So looking at that other white spots as well. Why can female cats be black and orange and males can't? Well, the short answer is females have two X chromosomes, and they shut off one or the other X depending on the cell. And so depending on which one's shut off, the other one will be turned on and either produce orange or black fur coloring. The gene for a trait is on, if it's on an X chromosome, there can be different results for that trait in males and females. Any trait that is made by a gene on the X chromosome is called, well, let's go find out. So let's go on to patient history. Let's investigate 3 different genetic mutations that could cause Calix to have calico fur. So it could be a point mutation where one nucleotide pair messes up in the copying of DNA, in DNA replication. When we copy our DNA, sometimes mistakes are made. And that's called, if it's just happened in one nucleotide pair, one place where the base pairs meet up, that's just called a point mutation because it only happens at one point. In one of the hypotheses here is that a point mutation caused the orange allele to become a black allele in Calix. So the mutated cell in Calix would now be black. A point mutation in an X chromosome could change some of his orange alleles into black alleles. That's one of the hypotheses. This change in DNA could cause the cell's fur color to go from orange to black. When the cell divides, its daughter cells also have the black allele. The mutated cells, well, can't read it now. Daughter cells with orange alleles, daughter cells with black alleles. That could have been the case. There's another mutation called chromosomal rearrangement. Let's watch this. Where a chromosome does crossover with another chromosome and loses part of itself. So we can look at these two chromosomes. They're crossing over, and part of one chromosome gets stuck to the other, so there's an extra piece. And this chromosome kind of gets gypped. It doesn't have as much DNA. It's shorter. That's called chromosomal rearrangement, when you have this extra little piece. And that's one way Calix could have, he could have gotten an extra gene for color, and so he got an extra allele. Maybe he got orange and black, depending on where that gene is. Calix could have inherited an X chromosome with an extra fur color allele. The X chromosome could have both orange and black fur color alleles. This would cause Calix to have patches of orange and black fur if he had chromosomal rearrangement. In which parent could chromosomal rearrangement have made an X chromosome with two fur color alleles? Do X and Y cross over in the father? Only mom, because mom is the only one out of mom and dad who has two X chromosomes. The next hypothesis here is non-disjunction, which means you get an extra chromosome, and this happens in humans. Non-disjunction is a mutation where chromosomes do not separate correctly. This causes one daughter cell to gain a chromosome and the other to lose a chromosome. Here's an example of non-disjunction. Those things are separating at that metaphase plate, but they don't separate quite right. And one cell ends up with an extra chromosome. You can see two of these have 3 and 2 of these only have 1, so something got mixed up. And that happens in humans. If the sex chromosome fails to separate, a male cat can be born with an extra X chromosome. He would be XXY instead of XY. One X chromosome could have the allele for orange fur, the other one could have the allele for black fur. X inactivation would cause the cat to have cells that are either orange or black alleles. This would result in patches of both fur colors. So mom could have 2 X's because things didn't separate right in meiosis, and dad could contribute a Y, and so the resulting cat is a male cat because it has a Y chromosome, but it has 2 X's. And in humans, that's called Klinefelter syndrome. Predict how each mutation would affect the mass of DNA in Calix's cells. Well, if only one point in the DNA gets changed, like a base pair gets changed out, that's not going to affect the mass of the DNA. So that's going to stay the same. I'll let you answer the next 2, but I'll give you big hints here. Chromosomal rearrangement is where you get an extra piece of DNA. If you have extra DNA, you have an extra piece, that's going to be extra mass. So what's that going to do to the total mass of DNA? And if you get a whole extra chromosome, instead of 1, you have 2, how is that going to affect your mass? You get a whole extra chromosome, that's more mass. So what's that going to do to the total mass of DNA? I'll let you answer those. So let's go back to our lab sheet here for a moment. Any trait that's made on a gene on the X chromosome is called an X-linked trait, or you could say a sex-linked trait. Explain how big O, little o female cats have some orange patches and some black patches because of X inactivation. Remember, in any female that has 2 X's, one of the X's gets shut off in any particular cell, but it's random which X gets shut off. So sometimes the big O X will get shut off, and sometimes the little o X will get shut off. If big O gets shut off, the cells are black. If little o gets shut off, the cells are orange. List 3 main differences between mitosis and meiosis. We have talked a lot, and there are a lot of good differences here in the diagram, so I'm going to let you study that up close yourself. Let's go over the mutations that we've looked at so far. Explain in your own words how a point mutation, in other words, a mutation in just one of the base pairs in DNA, would cause something that's orange, an orange allele, to turn into black. Just some small mutation could happen. And 34 is about chromosomal rearrangement. That means you get an extra little piece of DNA on your chromosome. You could get an extra color allele, and that could result in orange and black. So explain how getting a little extra piece of X chromosome that's caused from a mistake in crossover in meiosis could create a cat that is both orange and black and male. 35 is talking about non-disjunction, where you get a whole extra X chromosome. So you need to explain how you get to an orange and black male cat who's XXY. How does X inactivation work in this situation to give you a male that is orange and black? If you need help figuring out how cats get to be orange and black, you can go look at question 28. Here's an excellent diagram that helps you remember what alleles you need to be orange and black. Why is a black male cat a good control cat to compare to Calix? Well, think about the basics of picking a control in an experiment. You want that control animal to be as close to the animal you're studying as possible. If you haven't figured it out, these two mutations increase. Let's pick a control here. So we'll need to do some experiments to measure the mass of DNA in Calix's cells. We'll want a control cat to see how much DNA is in the control cat. We'd probably want to pick the cat with the same sex, so we want a male cat. A calico male is an unusual cat that's too much like Calix, so we want to have kind of a more typical male cat. So we can choose this one. So the choice is identical to Calix in every way except for color. So our first experiment here, we've taken cells from Calix and the control cat. Scientists use a machine called a cytometer to measure the mass of DNA in the cell. Words that start with cyto refer to things in cells like cytoplasm, cytokinesis. Meter at the end of the word means a machine that measures. So cytometer means a machine that can measure things in cells. So we're going to put the control sample in here and have the cytometer measure the amount of DNA in Coco, or the control male cat's cells. So that's the amount of DNA in the control. Let's look at Calix's cells. So how much DNA in Calix's cells compared to the control? Well, it certainly looks like from the graph that Calix has more. So Calix's cells have more DNA. That bar is taller on the graph. It's got more femtograms of DNA. Femto is a very tiny amount, tiny fraction of a gram. Let's look at the DNA mass in more detail. The experiments show that Calix's cells had more DNA. To find out if this is abnormal, we'll compare the data to normal data for cats. So Angie is Calix's mom. She's orange with white patches. So this is Angie's data, Calix's mom. This is the normal range. She's 1520, and the normal is between 1515 and 1525. She's right in the middle, so let's call her normal. Tails, Calix's dad, is 1495, and normal for males, because they have an X chromosome that's a little bit smaller, so they have a little bit less mass of DNA in their cells. That's normal, because that Y chromosome is smaller. He is right in the middle of normal as well. 1495 is right between 1490 and 1500. Now let's look at Calix. Calix is 1535, and the normal range for a male is 1490 to 1500. He's way over 1500. He's at 1535. He even has more DNA than his mom. So he's not normal. He's very high. So let's go interpret our data here. Why is the normal range for mass of DNA in a male cat cell lower than for a female cat? Well, that Y chromosome is smaller than the X, and males are XY, so males are going to have a bit less DNA than females. But Calix is really high. He's even higher than his mom. So that means he must have gained some DNA. That is some data that weighs against the hypothesis of a point mutation, because a point mutation wouldn't change the DNA. In chromosomal rearrangement, some of the daughter cells gain parts of chromosomes or lose parts of chromosomes. A whole X chromosome is 40 femtograms. If Calix was 1495, just like his dad, and he gained 40 femtograms, 1495 plus 40 equals 1535. And I'm just going to write that in the box. 1495 femtograms from Calix's father would be normal for Calix, or expected. However, Calix has 1495 plus 40 femtograms for a total of 1,535 femtograms. So it's likely that he gained a whole extra X here. So now it's your turn to make some hypotheses. What do you think happened? Was it a point mutation? Was it chromosomal rearrangement where a chromosome got a little extra piece tacked onto it? Or was it non-disjunction where you get a whole extra X chromosome? You make your hypothesis and put it in Gizmos, and also you can put it in your lab. So here's your hypothesis. You'll get more X chromosome data and you'll look at it. You'll also look at Calix's karyotype, and you can explain how Calix's karyotype is unusual using the words X chromosome, Y chromosome, and normal karyotype. Then you get to explain how Calix's genotype results in the phenotype. So what alleles he has results in his fur color. And then you've got the case summary here. You can explain what all that DNA data means. We've talked about that. And you can do the writing wrap-up, which means you use all of these words in sentences. You don't have to use every word in a separate sentence. I encourage you to use several words in the same sentence and write a short paragraph. It can only be 3 or 4 sentences is kind of what I'm looking for, using all of those words. Thanks so much for watching. This is Dr. B signing off. Good luck on the rest of your lab.